哇！
类似于收获者。
Here is what Peking is going to show. This year, we focus on uranium, a radioactive and one of the highest numbered elements found in nature. Uranium exists naturally in very low concentration. When used carefully, uranium could benefit the world as industry material or fuel for nuclear power plants. Just 1 kilogram of uranium-235 can generate as much energy as 1,500 tons of coal. On the other hand, when losing control, its radiation and chemical hazard can lead to serious environmental issues and health effects. Liver, kidney, brain, and other systems could be affected. The uranium leakage of nuclear power plants may bring fear and trauma to more than thousands of people. And during uranium mining, a large amount of uranium may enter the environment and cause negative impact. So, to figure out what's the current treatment of uranium contaminations nowadays, this summer, we visited one of the institutions of nuclear industry as part of our human practice. And surprisingly, we found that their treatment for uranium contamination was just to bury deep with fresh soil and grow plants on it. It's time-consuming and inefficient. And based on literature researches, we found some other chemical, physical, or biological methods. But they are not effective as we think, and all have many drawbacks, like the cost, complexity, and lack of efficiency. Considering that, our team wondered, could we design a biomaterial to dissolve uranium economically, robustly, and efficiently? And here it is. At first, we designed the fusion proteins and transformed plasmid into bacteria for proteins expression. And with thickened peptides, the proteins could be secreted by bacteria. The proteins we chose consist of 2 monomers, which could cross-link with each other. And multiple monomers could combine together and form a polymer network. Uranium binding peptide, as the key part of our material, could specifically bind to uranium ions in wastewater. What's more, if we add a new module in the material, the whole polymer network could even be cleared from wastewater. To achieve this goal, our project should compose of at least 4 modules. First, we have to form the polymer network with monomers which could self-assemble. Second, we want all the monomers secreted by bacteria. And third, ensure our polymer network works both in lab environment and in field. Last but not least, we want all the uranium cleared from wastewater. And now, welcome Jenny to show how we achieve the first 2 modules of our project.
Thanks, Coron. Now let's move on to the first module, polymer network formation. First, what polymer network? Compared with scattered monomers, a polymer is a highly concentrated unit, thus facilitating collection and clearance. Moreover, the dense structure of the polymer could stabilize the key module, uranium binding protein, and increase the absorption efficiency. Then, how to construct a polymer network? Although the network seems complicated, it only consists of 2 monomers, whose key elements are SpyCatcher and SpyTag. Later, SpyCatcher will be simplified as B, while SpyTag as A. The lysine residue in SpyCatcher and the aspartic acid residue in SpyTag could form an isopeptide that is highly stable. Based on these 2 elements, we designed various monomers with different copy numbers of A and B. An A series will then fuse with the main module, uranium binding protein, or UBP. And all the monomers will then standardize and submitted to parts registry. Among all these combinations, considering the tertiary structure of a network and expression efficiency, we chose AAA, UBP, and BBB as the major backbones. To verify the feasibility of this combination, we tested by mixing AAA MRP with BBB. Just within a few minutes, a gel-like substance came into birth. To increase the specific surface area, we didn't lighter gel for absorption efficiency. So for subsequent experiments, we used a low concentration of AAA and BBB. SDS page proved that a polymer with high molecular weight was formed. Examining the results in detail, we could find the polymer is actually a mixture with various molecular weights. Then, is it possible to determine the weight distribution of the polymer? Yes, we did it with our software named SOP. Its function is to calculate the weight distribution of polymers based on Fourier theory. As shown here, it is a very complicated formula. But with our software, what you need to do is just input two parameters. First, F, the functional group number, and second, N, the molar concentration. Take AAA and BBB for example. Here, FA and FB equals 3. We just need to input the molar concentration for A SpyTag and B SpyCatcher. Just with a click, you can get the theoretical weight distribution of the polymer and use logarithmic coordinate to change the horizontal bond. Then, how accurate is the theoretical prediction? Comparing the simulation with experiment results, we found that whether for equivalent reaction or inequivalent reaction, these two results fit quite well. Furthermore, we tested the dynamic properties of the polymer network. As shown in the graph, our polymer network is quite robust to a wide range of temperature and pH values. Moreover, just within 1 hour, almost all the reactants turn to polymer, which means polymer formation is quite fast. Now we have constructed a polymer network with a lot of desired properties. And to produce the monomers inside, we want to engineer bacteria to secrete them. So here comes the second module, secretion. Secretion is to transport target proteins from across the membrane. Compared with the tedious work of protein purification, secretion could simplify the whole process, making it convenient and time-saving. Moreover, secretion could achieve continuous production, rendering our material more economical. As secretion itself is not an easy job, we found a short peptide named signal peptide, which can guide transportation across the membrane. So we fuse the signal peptides to the N-terminus of target proteins. As the secretion efficiency for signal peptides is unpredictable, we built a signal peptide library to screen for the best candidate. From previous studies, we selected 9 signal peptides and fused them to 2 target proteins. So far, we have constructed all the combinations and submitted more than 40 parts. Then we tested the secretion levels. If secretion doesn't occur, all the proteins will be restrained in the cytoplasm. Otherwise, they will be identified in the medium. So we separated the cytoplasmic components from the medium by means of centrifugation, and then a Western blot was conducted. From the results, we could see both of our target proteins can be successfully secreted. As shown in the combined results, various signal peptides, including OMPA, LT2B, and PHOA, can all secrete the target proteins. Up to now, we have achieved a secretable polymer network. Then, what about the main function, uranium adsorption? What is the main part for that function? My teammate Simon will introduce it in detail.
Thank you, Jenny. Fortunately, we indeed found a special protein called superuranium binding protein, simplified as you'll hear later on. It has a metal binding site with extremely high affinity for uranium due to a negatively charged pocket and a particular binding configuration. As shown in the histogram, most metal ions were not able to outcompete uranium, even at a million-fold excess. Also, according to the carbonate competition assay, SUP showed a dissociation rate of 7.4 femtomole per liter with uranium, which means our key part for uranium adsorption is of high affinity as well as high efficiency. In order to evaluate the adsorption capacity, a separation assay was performed in advance. The mixture was added to a filter. After centrifugation, proteins with a binding uranium would be blocked at the top, while the residue part would be done to the collection tube. And we defined the adsorption capacity as shown here. In order to quantify the concentration of the remaining uranium, we managed to find the indicator horizontal 3. It could bind with uranium to form a stable chelate with specific absorbance states at 652 nanometer and 800 nanometer. With the concentration gradient, we could get a series of absorbance concentration values. And the uranium concentration showed a relatively high linear correlation with absorbance. In order to meet our detection to a limit, SAP max was applied for low uranium concentration detection, with the detection magnitude even down to nanomole. Then we made a series of quantified experiments to evaluate the adsorption capacity for our polymer network as well as its backbones. For the equivalent more-rich reactions, the adsorption capacity was 87% for AAA SUP. Moreover, what we really care about was when AAA SUP cross-linked this BBB and form a polymer network. Could the adsorption capacity be increased even more? And yes, the adsorption capacity was even increased from 87% to 96%, which means each gram of our polymer network could adsorb up to 3.5 milligram of uranium. What's more, we also evaluated our polymer network's performance in natural environments. We simulated the uranium-contaminated lake water and seawater by adding a uranium concentration of 10 micromole. For lake water, the adsorption capacity was 67%, and for seawater collected from seashore, it's even up to 83%. Noting that the ocean uranium abundance is only 30 nanomole, much lower than our previous experimental concentration. So could our polymer network still have the adsorption capacity at such a low concentration remains a question. Thus, a uranium concentration down to 30 nanomole was applied to the evaluation. For TBS buffer, the adsorption capacity was 47%. And we even got our sample of seawater directly from seashore of Bohai Sea, part of Pacific Ocean. And surprisingly, only within 1 minute reaction, up to 33% of uranium could be adsorbed by our polymer network. So far, we successfully demonstrated that our polymer network has an extremely high uranium adsorption limit capacity as well as a low adsorption limit. However, we don't want to leave our polymer network in the environment. So, how to solve this problem? My teammate Becker will tell you the solution.
Thanks. As mentioned, the next problem we need to solve is how to clear the polymer network from the environment after using. From this picture, we can see that all the proteins in the polymer network have been cross-linked with each other. So, if there is one component which can be easily recovered, the whole polymer network will also be recovered along with it. Now, the question is, what can this component be? After thorough consideration, we thought MSA might be the best choice. MSA is a rationally designed monomeric version of streptavidin, which can maintain a stable tertiary structure without oligomerization. Just like normal streptavidin, MSA can also bind to biotin with high affinity, which is one of the strongest non-covalent interactions known in nature. So it would be very easy to recover it by biotinylated magnetic beads. To achieve the clearance of the whole polymer network, we first fuse the MSA with SpyTag module so that it can be incorporated into the polymer network. And the fishhook is exactly the biotinylated beads and a magnet. Using these tools, the whole polymer network will be cleared easily. To verify our design, we measured the concentration of purified AAA MSA after bead treatment and calculated the amount of decreased portion. From this histogram, we can see that 86% of the AAA MSA could be cleared. On the contrary, only 21% of AAA was cleared. This result demonstrated that MSA is still able to work with high efficiency, even though it was fused with SpyTag module. Then, we added SpyCatcher to the system and incubated them for 1 hour before bead treatment. Just as expected, a large portion of proteins were eliminated by the beads. So, we concluded that the polymer network could indeed be cleared by incorporating MSA. Therefore, we assume that the super uranyl binding protein can definitely also be cleared, but the efficiency should still be evaluated by experiments. Next, my teammate Coral will show you the results when the above modules were combined together.
Thank you, Michael. As all the modules are ready, what if we light them up, combine the modules to one? How well will the entire scheme perform? In the integrated experiment, we tested the clearance efficiency of our polymer network in different conditions, and as expected, our Uranium Reaper shows excellent performance both in lab environment and in field. The efficiency could up to 60%, and just 1 gram number of proteins could absorb uranium ions from more than 10 liters of uranium-containing wastewater. And we believe that after optimization, our strategy could also be applied in industry. The monomers could be secreted by bacteria reproductively in the fermenter, and the polymer network could treat uranium contaminations with high selectivity and efficiency. Also, it is easy to be cleared from wastewater by bioencoded material. Comparing with other treatments using the phytoremediation requires a long-term commitment for at least 30 days, and titanium dioxide adsorption is limited at pH lower than 6 and temperature higher than 90 degrees Celsius, even though it is much more efficient. By contrast, our Uranium Reaper could absorb 60% of uranium ions within just 2 hours in moderate conditions, which suggests it is efficient and robust. But we want to do more. Apart from uranium, there were many other heavy metal contaminations. So you may ask, what if we replace SOP with other heavy metal binding proteins? The strategy for decontaminations still work? And we actually did that. From the candidates, we focused on 2 main heavy metal ions, lead and cadmium, both are detrimental for human health and environment. We fused lead binding protein and cadmium binding protein, replacing SOP to the backbone of our polymer network. And from the encouraging result, we can say the Uranium Reaper can transform into lead Reaper, cadmium Reaper, mercury Reaper, and so on. As you have already figured out, just like MET binding protein, other functional units could also be applied in our polymer network. And the combination among this functional unit could provide our polymer network with a great potential for further applications. Our project can extend to more than heavy metal decontaminations. Apart from concentrating on experiment, our team did a lot in human practice. We visited institutions of the nuclear industry and did field research, as mentioned before. Also, we sampled from seaside and coal mines. By handing out the questionnaires to the public, we found people knew little about the impact of uranium contamination and the ways of protection. Through consulting to professors in special field in Peking University, we designed a brochure and handed them out to the public. In addition, we held a science summer camp to popularize iGEM and synthetic biology. Doing social work out of lab lets us know that practice is an important criteria for testing truth. Meanwhile, our team paid much attention to collaborations, to share ideas and promote each other. At last, we want to thank our teammates for their contributions. With you guys, I had a wonderful summer. And we want to thank our instructors and advisors for their generous help to our project. Above all, that is our project, Uranium Reaper. Thank you. Thank you all.
I had a question about the signal peptide that you added. Did you ever investigate the cleavage efficiency? In your slide, you're showing that the signal peptide is cleaved off. Do you have any data about the efficiency of that cleavage?
The construction of the signal peptide library is not based on the mechanism. It's based on the previous literature's performance of the signal peptide. Because currently proteins that are less hydrophobic, highly hydrophobic, or fast, rapidly folded. Rapidly folded are directed by 3 distinct pathways, but we are not sure about our AAA MSA or AAA SUP or triple SpyCatcher's folding state, so we cannot directly rational design a signal peptide for them. But we can use the previous literature's results, such as OMPA, LT2B, and others. We have applied the Western blot and ELISA results to test the signal peptide's efficiency. And here, in our Western blot result, you see this triple SpyCatcher. You see in the median control group show no signal, show no protein, because in our control group we express proteins without the signal peptides. So, and in the OMPA, LT2B, or PHOA, or PLB group, we express proteins with signal peptides.
And actually, we did some experiments to get the quantitative results. And from ELISA test, we tested the different signal peptides secretion efficiency for BBB. And the most efficient one is we can reach at least 15 milligram per liter, the secretion value. But actually, we failed to get the results for AAA UVP, so we didn't put the results on our slides.
Thank you.
Hi, so I imagine you would want to use this in seas or soil eventually when the whole thing comes together. So right now the clearance is based on magnetic beads. How do you plan on implementing this in the field?
In the future, we want to put the bacteria in the fermenter, but not in the real ocean or in the soil. So we just produce our proteins in the fermenter and then concentrate the proteins and we collected the wastewater and put our proteins in it. And we will not use magnetic beads, and we will use other biotininated materials such as biotininated resins or some other materials that could have a high efficiency for our clearance.
So you essentially want to take contaminated water or soil and pass it through a fermenter where it gets cleared and then passed back to wherever it belongs. That's the plan?
No, not a ferment. The fermenters are used to raise the bacteria. And we will collect the polluted water in a restricted box or some room, and we mix our proteins with the contaminated water together to do the adsorption work. Then the cleared water will be separated with proteins. And yeah.
Thank you.
Hello, thanks for a great talk. I had a question regarding the polymer network itself and the uranyl binding. Do you think the uranyl can access the center of the polymer network effectively? And do you think that the density of the polymer network can be a limitation as to how much uranyl that the network can actually bind?
Actually, in order to increase our adsorption capacity, we made a sol-like matter rather than a gel-like matter. So its density is relatively low, so that the uranyl could come to everywhere of our polymer network, so it would not be blocked if the polymer has a high density.
Thank you.
Okay, 5 minutes is up. Thank you very much.